everyone, and welcome to today's webinar on smart environmental monitoring programs. We are excited to have you here with us. My name is Jessica, and I am the Director of Marketing and Sales here at AMTech. I will be moderating today's webinar. First off, we encourage everyone to participate by asking questions throughout the presentation. You can submit questions by clicking on Q&A at the bottom of your screen, and we will save some time at the end to go over all the questions that came through. We will also be recording today's webinar, which will be made available on our website. To start off, for those of you who don't know, AMTech is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing, environmental monitoring, and special research projects such as shelf life, challenge, validation, and investigation studies. Today's webinar will be led by Morgan Young. Morgan is a microbiologist with several years experience in food safety and pathogen research and industry experience, creating and running successful environmental monitoring programs. We are also joined today by PhD scientist, Dr. Florence Wu. Dr. Wu has extensive experience in pathogen detection, microbial contamination control, and environmental monitoring sampling strategies. And with that, let's get started. Morgan?
Thank you, Jess. All right, today's webinar, the topic is on smart environmental monitoring plans. Food companies are required to develop and carry out environmental monitoring plans, but how good is your EMP sampling plan? Is it meaningful, statistically sound, and efficient? Today, we will discuss how to avoid pitfalls, increase efficiency, and take meaningful samples. These are the main topics of today's webinar. We will focus on two aspects of an EMP. One, key elements of a sampling plan, and two, how to maximize its efficiency, that is, how to make it smart. It is important to note that this webinar is not intended to discuss all the basic elements of an EMP. Rather, we will focus on ways to sample and test indicators that are really relevant to your product and your process.
Thank you, Morgan. First, we need to define how to define what is the smartness. The word itself means intelligence, intuition, and ability to understand. In my opinion, a smart EMP provides intelligent insight to meet your food safety goals. It is not really about regulatory compliance, although we need to do that, and also not about passing audit, also we need to do that. But as a part of your food safety plan, the ultimate goal is to ensure that the food you produce for consumers are wholesome and do no harm. Being smart, the first question you would ask yourself is why? Why do we need to do environmental monitoring? What purpose does it serve? There are three reasons. One is the reality of life. Second is needs of doing business. And third is self-serving. So the reality of life is that we must be compliant to regulations. For example, FDA has a requirement for preventive control. And the need of doing business is that we must meet customers' specifications and we must pass the audit if we want to have any customers. However, I believe that a company's real purpose of doing environmental monitoring should be self-serving. A good environmental monitoring plan can help us to validate environmental control and sanitation. It can help us to validate our food safety plan, and ultimately it helps us to protect our brand name. Nowadays, bad news can reach millions of people in few minutes. To produce wholesome foods that provide nutrition to people and do not cause illness is fundamentally important to a food company's business. Regulations and FDA expectations can help us to develop a proper EMP by serving as a framework of standard, and that including identify the risk that is relevant to the food you manufacture, process, pack, or hold. Determine which risk need to be controlled to prevent foodborne illness. Understand how to control the risk whether it's in plant or in supply chain. Make sure that those risks are being controlled. And finally, track the system that's working every day. Again, the main purpose of environmental monitoring should be to protect your food, the public health, and your brand name. The goal of environmental monitoring, therefore, is to prevent cross-contamination. Let's look at the facility contamination risk. Many factors can contribute to facility contamination. That include ingredients, machinery, people who work there, the environment itself, your process, and the history of the facility and the constructions that the facility is undergo or has been done in the past. So it is not necessarily, it's not only necessary, but very important to have an environmental monitoring plan so you can keep all those risks under control. The smart way to develop an environmental monitoring plan is that you and your team go through the entire process. Firstly, learn and understand the government or trade associations guidelines so you know the scope and what are the relevant elements of EMP. Secondly, perform a risk assessment to understand the risk of contamination that is specific to your ingredients, your process, your product, and your environment. This requires intimate understanding and the knowledge of your business that no one else has, and probably no one else cares more about it as much as you do. Thirdly, it is smart to collect the baseline data to demonstrate the microbial burden in your production facility and help you with the risk assessment. Instead of speculating, you can just perform some sampling, send a sample to a lab, obtain some background baseline data. It is the easiest thing to do and the low-cost effort to understand your environment. Hopefully, your EMP is developed by following the guidelines, assessing the risk of contamination, and understanding the result of baseline data. A well-developed EMP should meet your overall food safety objectives. So what an EMP looks like? What are the major components? A complete EMP should have at least this component. The first is, again, risk assessment. And you need to present the result of your risk assessment for the reasoning of your sampling plan. Sampling plan is a component that in the EMP that need the most work on. The sampling plan should include when, where, and how often to sample and what to test. And you might also answer the question why. And that why should based on the result of your risk assessment. Acceptance criteria that define what are acceptable and what are considered out of spec should be in the EMP, whether it's a pathogen result or indicator enumeration. Corrective action is what you plan to do when you have out of spec testing result. And then finally, EMP should be reviewed and verified periodically to bring it up to date with your environment and your process. We'll look at this component in more details. Risk assessment is often dealt with a zoning concept. And the zoning concept will help you to determine your sampling plan. So in addition to analyze all the risk factors can input into your environment, you can further classify your environment into areas. Areas is designated where the product is most vulnerable to microbial cross-contamination. so that a proper level of monitoring and hygiene can be assigned. And then underneath it, you need to define the zones. So each area is divided into zones that refer to the location of the swabbing points based on closeness to and impact on the product and the food contact surface. And then underneath that, you can design the sites. Sites are the actual environmental sampling locations with a zone that must be identified and mapped, and of course, as a guideline for performing the actual sampling. Here is a diagram of classification of zones that serves as an example. So zone 1 typically is product contact surface. That can include the slicers, peelers, fillers, hoppers, screens that in direct contact with the food, such as also like employees' hand, knives, racks, work tables. Zone 2 is normally non-product contact surface, but it's very close to zone 1. That can include the exterior under a framework of equipment that produce food, refrigeration unit, equipment housing, and the switches near the production line. And zone 3 are the other areas within the finished product room that it can include the air return covers, phones, hand trucks, forklifts, strings, and wheels. Zone 4 is the area outside of the production area, and that can include locker rooms, cafeteria, hallways, loading dock, and maintenance area. If the zones are classified based on their risk level, then they should not be treated as equal in your EMP. So normally, in general, we see that samples are taken more frequently and in larger number and test for more in zone 1, zone 2, and less so in zone 3 and zone 4. And performance standards are also more stringent in zone 1 than in other zones. Here is an example of sampling plan according to the zoning concept. It is really just an example for a small sized company. For example, like zone 1 usually is the food contact surface. You can do daily test of ATP or indicators or both. And then you can do weekly pathogen testing, or if you have an allergen management program, you can test for allergen. The number of samples, of course, depends on how many production lines. And it is recommended a minimum 5 samples per line. And in zone 2 is the area that adjacent to food contact surface. And you can test ATP for on daily basis and indicators or pathogens or both on weekly basis. And the number of samples depends on your operation. 10 to 15 seems to be a number throughout there. But you need to decide it for yourself. And zone 3 and zone 4 are sampled less frequently, and you might want to test for pathogen alone, and the number of samples can be reduced from the number from zone 2. Again, this is a really simplified example of sampling plan.
Thank you, Florence. That is a good but simplified environmental sampling plan. In real world situations, companies of different sizes, they have different risks and different processes. So an EMP should be customized to each individual company. However, in general, an environmental monitoring plan must be scientifically valid. It must identify the test microorganism or microorganisms that will be tested for. It must identify the locations from which samples will be collected and the number of sites that are to be tested during routine environmental monitoring. It must identify the timing and frequency for collecting and testing samples. It must identify the tests that are to be conducted, including the analytical methods that will be used to perform these tests. It must identify the laboratory who will be conducting these testings. And then it also should include corrective action procedures if there is some sort of issue that arises from this testing. When fully implemented, the EMP should create a baseline of the microbiological assessment of a plant's environment. This will identify the potential sources of microbial contamination and possible vectors that may harbor or spread contamination. An environmental monitoring plan also provides verification of the effectiveness of sanitation practices. It will also provide verification of the effectiveness of procedures used to segregate and control traffic. This can include personnel and equipment that are moving throughout the facility. It will also provide data to correct problem areas before they pose a risk. And that's always what we want to do, is catch it before it poses any risk to our product. And then lastly, it will provide trending data to modify and update programs in response to results and observations. So you will need a few tools in your environmental monitoring program, and each of these do have a place in your program. We will have visual inspection. We'll also use ATP bioluminescence. We'll use microbiological testing. And then there are also other technologies that we can use, such as DNA sequencing or whole genome sequencing. Today, we'll touch a little bit on visual inspection, a little bit on ATP, but we're mostly going to focus on this microbiological testing. So, of course, to begin, a little bit about visual inspection. When an environmental contamination issue arises, one of the first things needed is a visual inspection and a walkthrough of the facility. You're going to inspect harborage sites, intensify your cleaning. You're going to watch product handling practices. You're going to watch traffic flow of people and product. You're going to watch cleaning by the sanitation crew. You're going to examine for any structural damages that might be in your facility. You're also going to want to ask assistance from your line operators to really understand the process that your product goes through each day. And then you'll also examine product flow and assess contamination risk. As you can see in this little picture of a small facility that we have, you know, if you were to have an issue that arises underneath this conveyor, for example, you're going to really watch the process through sanitation, through production, and that's all going to require visual inspection. Next, as I mentioned, we're going to talk a little bit about ATP and how it can be used in an environmental monitoring program. So what exactly is ATP and what does it test? ATP is adenosine triphosphate, which is the energy molecule in cells. ATP is present in all organic matter. ATP is read in RLUs, which stands for relative light units. So the substrate and enzyme complex in cells combines with ATP to produce light. Luciferin and luciferase are what connect or combine with that ATP and produces light. So the more ATP there is, the more light will be produced. Luminometers like this one that is pictured here are used to quantify the amount of light produced from a swab sample that is taken. This allows for acceptable limits to be set for your process and enables data trending over a period of time that you can use later on. So when exactly should we sample? When it comes to sampling, of course, this is a huge question. So when, where, how much do we sample? And like I said, right now we'll discuss when exactly do we sample. So during pre-op, in general, this data should be easier to interpret. You're less likely to yield a positive result. And then also this is the time where we will be able to identify sanitation weaknesses. During operation, you're more likely to yield positive results here. Data from operation will provide information on the spread of the target pathogen during processing. Positive sample site may not be the site where the pathogen survives. So it will require pre-op follow-up afterwards to identify that pathogen source or niche. So during post-sanitation, this will also help to really evaluate your sanitation effectiveness. And something that is very important to note is that when you are sampling during operation, the FDA does recommend testing 3 to 4 hours after the start of production. Okay, so there are many different sampling techniques. We won't get too much into detail with these today, but do make sure to check out the Amtech Laboratories YouTube channel, and you can watch all of our sampling videos there to understand exactly how to take an air sample, a swab sample, a water sample. We have a lot of different videos there for your access. There we go. Okay, so let's talk about testing now. So for pathogen testing, when do we test for pathogens? So for the zone 1, you routinely want to test for this. This is what the FDA recommends. If you do happen to get a presumptive positive, then of course you want to continue testing for pathogens investigationally. It will also be part of your root cause analysis to do more testing. Anytime there is a new line, you're going to want to test your zone 1 for pathogens, Listeria and Salmonella. Anytime that there is a construction or any major event in your facility, you're also going to want to test for pathogens. Anytime you have a positive in a food sample, or there may be a recall linked to your product, you're going to have to test zone 1 for pathogens. And anytime that you do have a positive in zone 2, 3, or 4, it's a good idea to also test your zone 1 for pathogens. And then anytime, of course, that you're having troubling trends from any of these other zones or in your facility, it's a good idea to test zone 1 for pathogens. For zones 2, 3, and 4, of course, we're going to routinely want to test those zones for pathogens. The pathogens that, like I mentioned before, we should be testing for our Salmonella and Listeria monocytogenes, possibly E. coli, depending on your process and your product. Of course, as Florence mentioned before, you really, when you're creating an environmental monitoring program, you're going to want to evaluate your risk at your facility. And then, of course, anytime you need to do an investigation or you're doing root cause analysis for a positive, you're going to want to test zones 2, 3, and 4 as the vectoring process. So talking a little bit about the pathogens that we should be testing for. So of course, everyone knows Salmonella. It's responsible for 94% of low moisture food outbreaks. We've heard of, chocolate, infant formula, peanut butter outbreaks with Salmonella. Salmonella, it survives heat and dry conditions. So if you have a facility where you have dry conditions or you have a heat process, you definitely do want to still test all of your areas for Salmonella. Harborage for Salmonella is where foods and dust accumulate and it might be a bit harder to clean. Salmonella can escape the dry cleaning process. So you definitely want to, test for Salmonella to really validate that your sanitation process is working. And then Salmonella can actually grow in areas that get wet and contaminate the food contact surfaces. So even in a wet facility, you definitely want to test for Salmonella. Next, we'll discuss Listeria species. So Listeria species includes 6 species and are found commonly in soil. El mono, of course, everyone knows that. That's the human pathogen. Listeria like wet and cold areas of the facility. So think of your drains, your chillers, refrigerated areas. That's where you're most likely to find your Salmonella. Harborage sites include areas where food and water are present. So if you have any standing water areas in your facility, like cracks or hard to clean areas, those are definitely where you want to focus on Listeria testing. So Listeria can grow and form a biofilm on processing equipment, and this makes it very difficult to remove and to clean. And of course, once a biofilm is formed, it's very easy for it to spread and then potentially contaminate your product. So good sanitation and environmental control are critical for the control of Salmonella. And then, of course, as you can see here, Listeria's prevalence in RTE, ready-to-eat foods, is 0.17 to 4.7%. So next we'll talk about E. coli. So of course, Listeria and Salmonella are the most common that you will test for in your facility. But of course, like I mentioned before, we might want to test for E. coli if your process, suggests that you need to do that. So E. coli does survive well in the environment. Of course, we all know about the history with E. coli in produce. It is pH resistant. So of course, with fruit juices, anything that might have, be more higher acid, you have to think about E. coli there. So it has been isolated from food contact surfaces, and it can survive in low moisture foods like flour. All right, next we'll talk about our indicators. Zone 1 sites are typically tested for aerobic bacteria, coliforms, E. coli, Enterobacteriaceae, yeast and mold, and these are usually tested pre-operationally before sanitizing and prior to startup of your production line. And so this will help to measure your cleaning effectiveness. The effectiveness of general cleaning and sanitation can also be determined rapidly using ATP testing. A large number of sites can be evaluated very quickly with ATP. So either a 2-class, which would be pass fail, or a 3-class, which would be acceptable, caution, or fail. Sampling plan, as described in a previous webinar, can be used for this. So if allergens are concerned, you can also use a rapid protein assay, and that can be conducted in about 15 minutes. And when using a quantitative indicator such as aerobic plate counts, performance target or acceptance limit should be established. All right, to discuss a little bit about some specific indicators, aerobic plate count, or APC, includes bacteria that are aerobic, mesophilic, and they are grown on standard agar. APC can be used to evaluate sanitation methods, adhere to GMPs, determine organoleptic acceptability, evaluate shelf life, identify spoilage in your product, and to evaluate conditions during processing, storage, and handling. Enterobacteriaceae. So Enterobacteriaceae are a family of bacteria that are gram-negative facultative anaerobes. Notable pathogens in this family include pathogenic E. coli, Salmonella, and Shigella. And many species from this family are commonly found in the gut microbiota of mammals, and it can be found on plant material. And so Enterobacteriaceae is usually tested as an indicator for Salmonella. Next, we'll discuss a little bit about coliforms and generic E. coli. So coliforms are gram-negative, non-spore-forming rods that ferment lactose to acid and gas within 48 hours at 35 degrees Celsius. Fecal coliforms ferment lactose to acid within 48 hours at 44.5 degrees Celsius. And E. coli is an indicator for fecal contamination. Some E. coli are pathogenic. For example, E. coli 0157H7 that we are all very familiar with. So coliforms and E. coli, as I mentioned, those are usually used to indicate fecal contamination in your facility. Next, we'll discuss a little bit about yeast and mold. So yeast and mold are indicators of moisture. Yeast and mold can be used in our testing plans to evaluate food spoilage and decomposition, to determine shelf life, to evaluate the adherence to GMPs, and then also to evaluate processing and handling conditions. All right, so it is very important to establish performance targets in your operation. These performance targets will help you to evaluate the effectiveness of your sanitation program. Different operations will have different targets. Baseline testing is important to establish your targets. So this chart here is just an example of a possible scenario for an operation. It's a good kind of base point if you're just starting out, but of course you're going to need to use your baseline data to truly determine what the most acceptable and unacceptable limits are for your facility.
Thank you, Morgan. You have discussed the sampling techniques and when and where and how often to sample and what to test. Now I would like to discuss how to establish baseline. For some people, they totally decide not to establish baseline, but I would argue that having a baseline can really help you to evaluate your sanitation performance. It is not really a difficult task. It's actually rather simple. The process will involve 3 steps. First is collect data, and second is analysis, and third is set limit. Collecting data, meaning you sample when the system is under control. You do not want to sample when the system is down or the system is under stress. You want to see under normal operation what's the microbial load can be. And you want to collect at least 20 samples to have a meaningful statistic analysis. And analysis will involve, of course, testing to get the result. And then you can use an Excel file for simple calculations. You can measure the mean, the standard deviation. To set the limits, there are different ways of setting the acceptance limit. You can use this using 2 criteria here. One is upper warning limit, which is the mean plus 2 standard deviation. And then you can also set upper control limit, which is mean plus 3 times of standard deviation. So this applies to the environment, maybe zone 1, maybe zone 2, that you want to collect quantitative result to evaluate your sanitation. If you decide to use other set of performance standard, as Morgan mentioned before, just I want to, after sanitation, I want the count to be less than 50 CFU per sample or less than 10 CFU per sample, that's fine. But for some other environment, have a baseline, have the acceptance limit is actually more effective in managing the environment. So there are different ways to do data trending. After you have the baseline data and you have used it to evaluate the performance of your sanitation program, and over a long period of time, you have accumulated a lot of data. What do you do with this data? The smart thing to do is to analyze data for trends, get more insight for your environment, and identify any patterns. It would be great if you can use your past data to predict future impact of sanitation or production activities. There are numerous resources for checking and trending EMP program. The simplest way is using Excel file. Excel is a popular software provided by Microsoft Office, which a lot of business have it anyway. Excel can do a lot of data analysis and produce chart that are easy to understand. But if you have a really large, complicated production area and involved hundreds and thousands of data points, many companies would have partially or fully automated software built into their sampling device. Some systems are highly automated, and both hardware and software are fully integrated into their overall food safety management system. There are also standalone software that tailored for managing environmental monitoring program. And finally, laboratory information management system can also be customized to track environmental testing data. If you work with a third-party lab, you can seek their help to generate a trending report. Here is an example of tracking data using Excel file. The chart on the left is the result of air exposure plate. The orange is aerobic plate count and the black is the mole count. You can see this is a quarterly sampling plan. It shows that the distribution of the count the number of each sample. It provides a trend, like what happened if you have elevated count and what is normal count should be. And then on the right is the result of out of specification sampling. This can be pathogen positive. It can be Listeria species, the indicator positive, and then you provide the number of positives in different month. See what happens. Why in January, February, and August there's elevated count? Does it associate with the increase of production volume? Does it associate with any particular construction event or seasonal change or ingredient change? So this gives you some clue that to look into the production activities and their impact onto your environment. Here's an example of a standalone EMP management software that is actually developed by Fremont, a company that has a state-of-the-art EMP program that including an electronic sampler, sampling vial, and software that has automatic data trending functions.
Thank you, Florence, for that information. So from my own experience, entering data and doing data trending manually, it can be very time consuming. I'm excited about Fremont's automatic sampling and data management system. So unfortunately, there is part of an EMP though that cannot be automated. That is when we need to deal with contaminations or out of spec results. When contamination in the facility is above the designated control limits, or when pathogens are found, investigational sampling will be started with much higher frequency than routine environmental sampling. Initially, contact and indirect food contact areas will be emphasized to find the contaminated sites and deep clean them. Then intensified sampling will spread to adjacent zones. The line will be stopped, equipment deconstructed for sampling and deep cleaning. It is essential to include perimeter zones and non-process areas in the sampling, since these areas can often be vectors for contamination due to traffic. The chart showing high-risk sampling sites is useful as a guide to start sampling. So what exactly do we do if we get that dreaded pathogen positive? So if it is detected on a food contact surface, then we should conduct comprehensive investigation. For example, an expanded root cause analysis. It is very important that we hold and test product for those production days that we did get that positive. We should examine equipment, going back to our visual inspection that tested positive, and the surrounding area. We should review and modify HACCP or food safety plans as needed. We should intensify our sampling and testing for a minimum of 3 consecutive days. We should test upstream and around the positive testing location. So this is also called a vector analysis. We should check maintenance records for any sort of major equipment changes that happened recently. And if needed, we should disassemble the equipment as well. We should interview and observe sanitation, maintenance, and production personnel to really get a full story of what was happening around that positive. And then we should review and modify production, maintenance, and sanitation procedures as needed. So further actions to be taken in responding to a positive pathogen results are to identify potential causes. An extensive review of traffic patterns, equipment layout, and adherence to personal hygiene procedure will help to identify these causes. If product or environmental positive, then you should reprocess, divert, or recall product as needed. We may need to stop production. And then, of course, you can always consult a food safety expert if you need some help with that as well. If food contact surfaces are negative for 3 consecutive days, then products from those days can be released, just from those 3 days that have been negative. A similar approach should be taken if a positive is found on a non-food contact surface. For these investigations, you will concentrate on zone 2. You'll, of course, conduct your root cause analysis and then determine if you need to disassemble equipment. When a positive is found, vector swabbing is conducted using a starburst pattern so that 360 degrees around the positive site is sampled. If additional positives are found in the vector sampling, then we will extend a vector sampling to determine the extent of contamination. Any item is suspect that is in the immediate area of the processing line. Any open cabinets, open panels, wheels or legs on equipment that are moving in and out of the area will also be suspect. Vector swabbing is used so that you don't forget to look up and above the product or below the product where niches and harborages can be found, and those pathogens love to live in those areas. Any drip, condensate, or airflow over the product is also suspect, including compressed gases. So what do recurring positive results mean? So detecting the organisms in several sampling locations during the same sampling period, it could indicate that there are some routine sanitation procedures that are not being followed correctly. It also could mean that you have some potential harborage sites in your facility. It also may indicate the presence of a resident strain. For example, Salmonella agonia, it persisted in the facility for 10 years. That's a pretty long time. If these are transient strains, this means that they are just passing through, and transient strains can be eliminated by effective sanitation that can prevent them from becoming resident strains. Recurring positive results do increase the risk of cross-contamination from zone 2 or zone to zone 1 or to your food. Okay, so sometimes corrective actions, they simply just don't work. In spite of every effort to monitor, sample, and correct environmental issues, including deep cleaning equipment and environments, production lines still test positive erratically for pathogens or pathogen indicators such as Listeria species. Such equipment should be segregated and removed until a solution is found. The facility should consider taking the equipment out of service if it cannot be cleaned properly. Thank you so much for your attention today. In conclusion, there are two main takeaways from our talk today. A food processing environment is ideal for the survival of pathogens. So of course, these key pathogens are Listeria, monocytogenes, and Salmonella. And then also our other takeaway is that EMPs are critical in a food safety plan. So virtually all processing plants need these EMPs, and then results need to be used for immediate corrective actions as well as long-term improvements. Thank you, guys.
All right, well, thank you everyone for joining us today. We do have a few questions that came in, so let me see if I can get those up here. The first question is, why should indicator testing be done before sanitizing?
Want me to take this one, Morgan?
Yeah, sure. Go ahead, Florence.
Yeah, so this is a good question. I think it depends on the purpose of your sampling, right? If you go just I want to kill all the bacteria on my food contact surface, and the result you want is when I need to bring the count into less than 10 CFU per sponge or per 100 square centimeter, then in that case, you probably don't need to do before. But if you want to evaluate your sanitizer and your sanitation techniques, whether they are effective, then you having a before data you can compare against too. And another thing is not all companies are created equal in terms of environmental risk. some companies, because their operation, mostly because the product they deal with, and for some surfaces, it's not possible to really every time you require to have less than 10. So you want, in that case, if the after testing, after sanitation, the number is high, you don't know whether the sanitation really work or not. So in this way, you want to collect before and after, and by comparison, you can evaluate the sanitation practice, whether they're effective or not.
Oops, I was on mute. Thank you, Florence. Next is, does a wash need to be performed after in-process swabbing is completed? So does a wash need to be performed after in-process swabbing is completed?
From my experience, it just kind of depends on where you are where you are swabbing. Of course, zone 2, 3, 4, typically not a wash after swabbing. You would just kind of want to evaluate your risk. If you are swabbing a zone 1, and then you determine that you should clean after that swabbing, you definitely can. But if you do not clean after your swabbing, then of course you would want to make sure that you are holding all of your product until you do get those results back. Florence, do you have anything to add to that?
I agree. I think for food contact surface zone 1, it doesn't hurt after you're swabbing, you clean up. Usually the swab material they provide normally come with neutralizer to neutralize any residue of sanitizers. It's supposed to be grass. It's supposed to pose no harm. However, if you have your food contact surface, it need to be maintained dry, need to be residue free, then I do suggest you clean or wash or clean after you do something, just the spot that you sampled. And then for zone 2, zone 3 or 4, that may not be necessary.
Okay. The next question is, why do we need to test, why should we test 3 consecutive days in a non-food contact surface? So why should we test 3 consecutive days on a non-food contact surface?
Yeah, that kind of goes back to your whole resident versus transient strain. So if you do have a resident strain, that initial sanitation right after your hit, it may inhibit the strain for a little bit, but then those resident strains, they will eventually come back. So it's one of those, you just kind of want to be persistent with it to really make sure that your sanitation program has gotten rid of and dealt with that problem.
Great. And just as a reminder for everyone, the webinar will be recorded and we'll make it available on our website this week. Next question is, to establish a baseline, what is the recommended time or what is considered a long time for collecting under controlled data points? So to establish a baseline, what's the recommended time or what is considered a long time for collecting under controlled data points?
Yeah, that's a good question. I only mentioned 20 data points for, it's kind of minimum for statistical analysis. The span between samples, can be however long or short as you see appropriate. For example, you can sample weekly. Then it will take you 20 weeks to accept a baseline. That is fine, unless if someone someone in your team or audit just wanted to know what is your acceptance criteria, and then you need to provide evidence of your acceptance criteria. Then you want to concentrate on sampling maybe daily, so you can get the 20 data point. And within like 20 days. I would suggest that the baseline established is not like a one-time thing. It is probably best to run through a year. So in some operations, especially like if you sample air, there will be seasonal changes. And then also if you switch out, ingredients or supplier, there can be changes out as well. So the longer time spent there, your baseline is more appropriate to your normal production.
Great, thank you, Florence. The next question is, does the acceptable limits need to be used in zone 2 and 3, or they work for zone 1 only?
It can certainly work on zone 2 and zone 3. For some companies, they don't sample zone 2 for indicator. And many companies definitely don't want to sample zone 3, zone 4 for enumeration, because it depends on what you enumerate, right? I see companies, they collect zone 3 and they often come with a high APC count, which is expected. With that high number, the trending almost not very useful. So zone 2 is an interesting area. I think that area may be more appropriate for indicator tracking, because zone 1, if you're comfortable with zone 1, I want to have less than 10 CFU per sample. Then zone 2, you can allow to have a range. Then when you have a number to trend and to analysis, then you can provide interesting insights. So to judge your sanitation.
Okay, thank you. The next question is, how often is it recommended to perform the air sampling testing? So how often is it recommended to perform the air sampling testing?
So I think that's really relevant to your production. For some companies do the monthly or quarterly or weekly. I think this is relevant to your product in the sense that if your product has specifications for yeast mold, then you should sample more frequently, because what is in the air can impact your product, especially if your product is prone to spoilage by yeast mold. And in collecting more data, in the air can definitely help you to decide, what activity generate more aerosols, can potentially, increase the chance for contamination. And same thing with bacteria. So, I would say, and let's do weekly in that cases. But if your product is the mold is not the issue, or airborne bacteria, there's no chance they can get into the product in some area, maybe not all area. So you want to differentiate in what part of production you want to do air sampling and how often you want to do air sampling based on that assessment.
Okay. The next question is, we currently use ATP swabs to verify the sanitation program. Is this not sufficient? So is it sufficient? We currently use ATP swabs to verify sanitation program. Is it sufficient?
Yeah, so that's a good question. While ATP swabs are definitely a quick indicator of organic matter that might be on surfaces, they can sometimes miss inhibited bacteria that can grow on your equipment when given the right environment. It is best to pair ATP swabs with APC or Enterobacteriaceae swabbing to truly evaluate the cleanliness of your equipment and to really see if there is a presence of bacteria, in your facility.
Okay. The next question is, I swab for Listeria in my refrigerated facility. Is it really necessary to also swab for Salmonella? So I swab for Listeria in my refrigerated facility. Is it also necessary to swab for Salmonella?
Yeah, so salmonella is it's kind of a tricky, we're finding out that it's a tricky pathogen. It has been proven to be pretty resistant. While it is difficult for salmonella to thrive in a cool and damp environment, it definitely can survive. We're finding that out. There have been several recalls in the past of salmonella in a refrigerated facility. So this is why it is important to still monitor for salmonella in a refrigerated facility.
Okay, thank you, Morgan. Next question is, how often is water testing recommended when it's used as an ingredient? So how often do you recommend testing water when it's used as an ingredient?
I would definitely suggest to, monitor that as often as you monitor your finished product. It's that can be a major source of contamination. It also can help you to know kind of the quality of your product as well.
And here's another good question. Do you recommend sampling for Listeria mono or for Listeria species, and why?
Okay, that's actually a pretty difficult question, right? Because when mono is, then it's pathogen. So I would say on regular basis, unless I'll tell you what are exceptions. On routine basis, just sample Listeria species. And then when you have presumptive positive, you decide whether to confirm it or not, and then just take action. So the action is, of course, corrective action, more sampling and more sanitation, verification, all that, EMP management program, right? Hopefully your EMP has that section, what do we do when we have positive. But the exceptions is, if your product detect the Listeria mono cytogenins, then you should, as a corrective action, you should sample your environment for Listeria mono cytogenins. And then if you repeatedly find Listeria species and you want to know what kind of risk you're having, I think that time, you should ask the lab to identify it all the way. So your practice can be heightened and it will trigger higher level of response.
Okay, thank you, Florence. Thank you so much, Morgan and Florence. With that, we are out of time for today. I want to thank everyone again for joining us. If we didn't get to your question, please feel free to shoot us an email at lab@amtech.com, and we'll be sure to get back to you with an answer. Again, that's on the screen, lab@amtech.com. We also encourage anyone who's interested in EMP assistance or testing to reach out so that we can schedule a meeting to discuss your needs. Or if you just want some more information and would like to talk to Morgan or Florence, that's perfectly fine too. Thank you again so much for attending, and we look forward to seeing you at our next webinar on Legionella. Thank you. Bye-bye.
Thank you. We all have a good
Thank
day.
you.
Bye-bye.